Iowa State University, and I'm in a research lab of Dr. Ann Bronikowski, and this is where I work when I'm not visiting you guys. So today, I want to show you a little bit about what I do and give you kind of an example of the type of research that I do. So what I'm going to show you today is how to extract DNA. So as you know from doing your understanding what life is, every organism has DNA in our cells. And so you have DNA in your tissues, I have DNA in my tissues. We want to be able to get that DNA out of our tissues and out of our cells. And so I'm going to show you how we do that using a blood sample. So here's a syringe where we took a blood sample. And what we want to do is in this blood it's full of cells. So here's an example of a cell. And we have our cell membrane and we have our nucleus. And in our nucleus is all this DNA. But there's all these other proteins that are in our cell that we need to get away from the DNA. So we want to get this DNA out of the cell, away from everything else. So we need to extract it from there, okay? I'm going to show you how we do that. So if we go from a blood sample, such as this, and we put it into a little tube, so as a molecular biologist, I'm always working with little tubes. And we have these tools. So this is a pipette, and we use it to measure out very small volumes. So you see that dial right there? Where it says,
we see that.
7 or 20. That means it's 20 microliters of volume is what it's going to measure out. So we're going to use this, and I put on this little tip. It's a disposable tip that I put onto the pipette, and it has a little filter in it so that I don't get any blood contaminating my pipette. And so I'm going to take some of this blood. I'm going to suck it up into the tip. And then I'm going to put it into this chemical buffer. And what this is going to do, it's going to, right here, I put it in there.
It looks like that's what I'm saying. It looks like spaghetti.
So now what this chemical is going to do is it's going to break open all the cells so that all these proteins and all the DNA can spill out. So we use this little machine here that we call Vortex, and it just mixes it up really well. Can you see how it's mixing it?
Yeah. Sort of. Yeah.
And then what I do is I would let this sit overnight so those chemicals can break down all those cells and spill everything out. And so in the morning when I come in, this is what it looks like. And so it's just kind of like a cell soup at this point. So everything's floating around in that liquid. We got the DNA, we got the proteins, we got the cell membranes. Now, how do you think we can separate that DNA away from all that other stuff that we don't want? You have any ideas?
You got it, guys?
Like a
exploded the cell, and now she just wants the DNA.
I want to know why it's brown.
So how would you get that out of there?
Around really fast so that all the other stuff goes to the side and the cells are in the middle.
You are so smart. We are going to use the centrifuge to spin that around, but first we got to add a chemical into there that's going to help us pull out. First thing we're going to do is pull out all the proteins that we don't want, so we can get rid of those first. And the chemical that we're going to use to do that is this one right here. Do you recognize that symbol? NaCl. What do you think that is?
NaC.
You remember what Na is? Sodium. And what's chlorine? Or, no, I just told you. Sodium chloride, which is what?
Salt.
Salt. Excellent. Very good. So this is a really salty water, essentially, to help us get rid of all those proteins. To our cell soup. And now we're going to use our vortex again to mix it up really good. It's spinning around really fast. Have you guys ever been in a silly silo? Yeah? What happens to you when you're in a silo?
Spin like crazy. You stick to the wall.
Oh, yes. This is the same thing that's going to happen in our little tube. That force of spinning around really fast is going to force all the proteins to go down into the bottom. And it spins really fast. It can hold a bunch of tubes. So we're going to put our little sample in there, and we're going to spin it really fast so all the proteins get cooled down into the bottom of the tube. It's 14,000 rotations per minute. That's pretty fast, huh? 14,000 rotations per minute.
That's really going.
So,
Yeah.
that's going to take a couple minutes. In the meantime, I want to show you this. What do you think this is?
Look at that piece of paper, guys. What's that look familiar to you?
It's a procedure.
It's my procedure, exactly. See, there I have my materials that I'm using. And then I have step-by-step directions as to how I do the extractions. So right here, number 5, that's where we added the salt. And number 6, now we're using the centrifuge to run it at 14,000 rotations per minute. And it says for 5 minutes. So just like you guys make procedures for your experiments, I make procedures for my experiment. Just like you guys have a lab notebook, except I have a different notebook for every single project that I work on. So if you can have 4, but I organize it kind of similar to you guys, and I have all these different tabs that tell me where in my notebook I should be putting procedures. And then every day that I work in the lab, I make a record of what I do, and I write down exactly what I do and what my results were for that day. So this is how scientists keep track of their experiments and their data to know what they've done in case somebody wants to repeat the experiment. They can do it exactly the same way. Okay, our tube is done. Let's see what's happened. All right. Look at that. What do you see?
It changed color.
Hold on, I'm going to kill the light.
Went to the bottom.
No, don't move it.
It all went to the bottom.
Yeah, I was like, I saw it.
Why did it turn brown?
So what is all that stuff at the bottom?
Like
Blood.
DNA. The DNA, yeah.
That's all the proteins. Remember, we had our cell soup, and now all the proteins are at the bottom. So where's the DNA?
At the bottom.
At the top, yeah.
It's in the liquid right here. So we're going to take off this liquid using the pipette, and then we can throw away all the protein. Okay, so we got a clean tube, and we're going to take off all this liquid. And we're going to leave all the proteins behind. Okay. Alright, so all of our DNA is in this liquid. And now we're going to throw away all the proteins that we don't want anymore.
Okay.
Alright, now we need to use a different chemical to get the DNA to precipitate out of the liquid. Not the alcohol you drink. It is 100% ethyl alcohol.
Is that a vodka?
No, that would kill you.
This will kill you if you drink it. But we're going to add this to our liquid, and what that's going to do is it's going to cause the DNA to condense upon itself, and it will precipitate out of this liquid. Okay, so let's watch. All right. Do you see that stuff in the middle there that's kind of like stringy?
Yeah.
Oh, yeah.
Yes, I see it.
That's the DNA.
That's that. Oh, wow.
That's the DNA.
Why do you? It looks like fizz. It looks like fizz, not really strain. It looks like. Why do you have gloves on?
Okay, how do you think we can get it out of there?
Suck up all the water.
Get that little thing that you like put in it and it sucks it up.
The pipette? Well, that's one way, but we might accidentally suck that up instead. What's another method that we could use that we've already used?
The filter.
The centrifuge?
Oh, yeah?
Sure. I don't remember what it's called.
The spin thing. The centrifuge.
So what we would normally do now is we would put this into the centrifuge, we would spin it around for about 10 minutes so that that DNA forms a pellet in the bottom of the cell, or in the bottom of the tube. But we don't have enough time for that, so I'm just going to show you an example of what it would look like if we did that. So if we spun that tube around for 10 minutes, what would happen is that DNA would form a pellet in the bottom.
That little dark spot.
That's the DNA. Now we could take off all that liquid, and the only thing that we would have left in this tube is pure DNA. There it is. That little speck in the bottom, that's pure DNA. Separated away from the cell, away from everything that's in the blood. Now that we have that, we can do anything we want. So this is the first step in working with DNA. So now this DNA could be used for forensic cases to figure out if somebody murdered somebody, or if they're a paternity analysis to see who the father of this individual is, or it could be used to study the whole genome. But this is the first step that any molecular biologist has to do to be able to work with the DNA.